Hello, everyone. Um, First of all, I would like to say thank you for hanging in here, both in person and virtually. I know it has been like a long week, an exciting one, but long, and you might be tired. But I hope you can stick with me for the following 20 minutes, and I hope you find this talk exciting. Um, so my name is Suzana Leles, and I am a postdoc working in the group of Naomi Levine. And the project that I will present today, it's also a collaboration with Noel Norris. And we have been interested in exploring phytoplankton responses to temperature changes. And throughout this week, we saw many interesting talks and results on this topic from Elena, David, Martina, using different modeling approaches and experimental manipulations, and that's all very exciting. So I hope here gives some contribution to this topic as well. And this work will be mainly focusing on molecular mechanisms that could explain phytoplankton trade-offs in response to temperature changes. So to get it started, as we all know, the oceans are getting warmer at an unprecedented rate, and it's very important to understand how biological communities will respond to temperature increases. And I am particularly interested on understanding how microbial will respond to warming, especially due to their role in the carbon cycle in the oceans. So if we look at this image here on the right, we can see the diverse microbial community that we can observe in the ocean. And something interesting about them is that they contribute both to primary production and respiration. So if we think about that, the balance between these two processes will determine if microbial communities, they are acting more as sources or sinks of CO2. So one question that we can ask ourselves is how this balance would change as temperatures increase in the oceans. And if we look at the literature, what we find is that actually theory predicts that microbes would capture less CO2 due to warming. And why is that? So before going into that, let's just remember the thermal performance curve. We have seen this before during this meeting, but basically we have metabolic rates increasing exponentially as a function of temperature up to a maximum. And after that, the metabolic rate is expected to decrease. So let's think here about a focused microbial population. And what we can do is that in the lab, it is possible to empirically derive these curves. And researchers have done that for both phototrophic and heterotrophic processes. And what they found is that heterotrophy is more sensitive to temperature than phototrophy. So basically what this means, if we look at this schematic here, is that the slope of the red curve is steeper than the slope of the blue curve because respiration is increasing at a faster rate than photosynthesis as temperatures increase. So let's keep this prediction in mind. And the second one that I would like to highlight here, and we are probably more familiar with this one, is that size is expected to decrease due to warming. And this is almost an universal response. It has been observed across different organisms, and it will also play an important role when we try to understand how changes in microbial communities due to warming will affect carbon cycling. So we were interested on these two predictions, specifically because the mechanisms behind them, they are not well understood. So that's the objective of this project, is try to understand better what is happening at the molecular level. So let's take a closer look at the temperature puzzle of phototrophs. So how to balance energy supply and demand to deal with warming? Well, we can imagine that actually phytoplankton will not just sit there and be relaxed when temperatures are increasing. And sorry, I love Mafalda, and I always try to find a way to add them to my talks. So if we think about phytoplankton, and especially first photosynthesis and how it is expected to change with temperature, we can think about two reactions in photosynthesis. We have photochemistry and we have rubisco. And while rubisco should increase its activity as temperatures increase, this is not true for photochemical reactions. They should be constant as temperatures change. So what we can see here is that at low temperatures, we have a surplus of energy because the photochemical capacity of the cell is higher than the capacity of rubisco. But as temperatures increase, these lines, they should tend to converge. And therefore, we have some energetic constraints in the cell. So this takes us to our first hypothesis, in which energetic constraints in photosynthesis would promote higher respiration, because the cell needs to find a way to acquire more energy. So now let's think about respiration. What is happening? We know that respiration should increase as a function of temperature. In recent transcriptomic studies, they have found some exciting results, because they show that not only respiration increases, but there is also an upregulation of fatty acid degradation at high temperatures. So we try to put this here in a schematic. What is going on within phytoplankton respiratory pathways? So we can imagine that at lower temperatures, because the cell has very low metabolic demands, maybe it can invest on a pathway that gives you a low energy yield, and it also has a low proteomic cost. But as temperatures increase and energy demand also increases, then the cell might need to switch to a pathway that can give a higher energy yield, but it might also have a higher proteomic cost. But can we observe such a switch? And what is interesting is that recent studies, they have shown that actually phytoplankton, they can store lipids during the day and burn these fat acids during the night. So this is something that has observed, and another maybe advantage for that is that lipids, they also they occupy less space in the cell compared to other carbon storage compounds. So this takes us to the second hypothesis of this study. So, and this would be that lipid degradation should be favored at higher temperatures, because lipids store more energy per mole of carbon, and they also can take up less space in the cell. So our goal here was to develop a model that could test this hypothesis. And we want to develop a model for a phytoplankton cell that can optimize proteome allocation to different functions to maximize its growth rate. And we do that at different temperatures. So I will walk you through this conceptual model here. So here you can see a phytoplankton cell, and we have different pools. In blue, we have different proteins, and in green, we have internal pools of nitrogen and carbon. So this cell takes up three external resources. So photosystems are responsible to transform light energy into chemical energy, and this energy will meet Rubisco demands and also other metabolic functions of the cell. Rubisco will fix inorganic carbon into carbohydrates, and transporters in the membrane, they will bring external nitrogen into the cell. And then internal nitrogen and internal carbon pools, they are used to make up ribosomes, which in turn are responsible to synthesize all proteins in the bottle, including itself. So carbohydrates, they can also be used in a respiratory pathway called here glycolysis and generate energy, or they are used in the lipid synthesis pathway, which must be used to build membrane lipids, or it can also be used in the lipid degradation pathway, which is also an option to generate energy. And glycolysis and lipid degradation here, they are described to meet dark respiration demands in the model. Now, our model, and something important about it, is that our solutions that I'm showing here today are all at steady state. So we are assuming balanced growth here, a population growing exponentially. And based on that, we can describe the mass balance equations of the different pools in the model. So this equation here shows the equation for the internal pools of nitrogen or carbon. So we have that the sum of all synthesis and degradation rates over a certain pool minus the dilution rate should be equal to 0, as we assume balanced growth. And all the synthesis and degradation rates, they take the form of Michaelis-Menten-like equation. And the mass balance equation for the proteins is just a special case of the first one here, because, as I said before, all proteins in the model, they are synthesized by the ribosomes, which is this flux here, BRIB. And what we are most interested on is on the values of the phi parameter, because these are the relative investments on the different proteins in the model. And basically, we are assuming that a certain fraction of this flux will be used to synthesize a given protein in the model. So the sum of all allocation factors should equal to 1. So in order to include temperature effects, we first assume that maximum reaction rates, they scales according to the Arrhenius equation. And remember, an important assumption in the model is that photosystems, they are assumed to be temperature independent. And finally, we represent protein damage due to high temperature, and we assume that this requires repair. So you can see here in the schematic that the photosystems, they will get damaged as temperatures increase, and the cell will need to invest in repair proteins to repair these damaged photosystems. So just summarizing the model, our objective function here, it is a constraint optimization problem, and our objective function is to maximize the growth rate. We will be optimizing the proteome allocation to different functions, and we can also optimize cell size by considering membrane constraints. So we assume cell volume is proportional to the surface area occupied by transporters and lipids in the membrane, and multiplied also by volume to surface area factor. And then we get also other emergent properties such as carbon use efficiency, the nitrogen to carbon stoichiometry of the cell, because we consider the different stoichiometry of different macromolecules. So that's very interesting that we can get some estimates for that, and also for chlorophyll content and total protein content. So what I would like to highlight here is that even though this model is simulating the molecular mechanisms and different proteins, we can still compare our simulations with data that is quickly obtained in the lab. So that's what I'm going to show you now, some model comparison against data. So here in these plots, the lines are our simulations and the symbols are the experimental data. So I'm showing you first on growth rate curves across different growth rate curves, thermal growth curves, and also carbon use efficiency of the cell. And the different colors here denote diatoms that evolved at different temperatures. And what we were interested was to see if our model was capturing the overall behavior. So we did not do an extensive parameter fitting here, but we were interested, and we saw that the growth curve, we can capture the overall pattern of the thermal curve, and we can also capture the decrease in the carbon use efficiency as temperatures get higher. So here you can see 3 different curves just to show a fast grower, intermediate grower, and a slower grower phytoplankton cell. So after doing that, what we are interested now is to try to investigate the molecular mechanisms that could explain these patterns. So let's have a look now at the modeling predictions. And what I will show you here are the optimized investments on the different proteins. So just remember that this is a fraction of the ribosome synthesis flux and how much it should be invested on the different proteins to maximize growth rate. So here you can see the relative investment as a function of temperature, and we are looking at photosynthesis to begin with. And what we find here is that as temperatures increase, the cell will invest more on photosystems to meet Rubisco demands. And if we can compare also our pool of photosystems with some data for chlorophyll obtained in the lab, that's very interesting that we can see we capture the increase of chlorophyll content per volume as temperatures increase. And our model also kind of captures an emergent, and this is something that emerges from the model, that the chlorophyll concentration should saturate at very high temperatures and eventually even get lower, but we don't capture the very low concentrations observed here in this study by Liang. And we can chat more about that if you are interested later. But it's nice to see that still the experimental data shows this increase in chlorophyll as temperatures increase. Meanwhile, metabolic demands of the cell are increasing, and also photosystems are getting damaged. So the cell also needs to invest on respiration and repair, given in blue here in this plot. And while the cell invests on these three pathways, photosystems, respiration, and repair, it ends up divesting resources from growth. And that's why we see the investment in ribosomes here decreasing as a function of temperature. And something else that is interesting with this model is that our model suggests a decrease in the N2C stoichiometry of the cell. And this is supported by the data, maybe perhaps at a lesser extent, but it's interesting to show this pattern. And if we look at these changes here, this is mainly because photosystems, they have less nitrogen per molecule compared to ribosomes. And that's what drives this pattern in the model, the decrease in the stoichiometry of the cell. So with that, we explored the first prediction, the molecular mechanism that could explain why respiration increases at a faster rate than photosynthesis as temperatures increase. So the second point that we were interested was that if we could predict changes in cell size based on membrane constraints. So what you can see here, it's our model simulating decreases in cell size as a function of temperature, and this is supported by the data. But what I would like to highlight is that this result was only possible if we make two assumptions. So first, we need to assume that transporters, they have a lower temperature sensitivity compared to at least one other metabolic process in the model. And this was crucial to see this trend. And another thing that could also help to explain decreases in cell size as a function of temperature are changes in the membrane composition. So because the cell can pack more membrane lipids at high temperature. If we don't make these assumptions, this is what the model would predict. So the model would predict actually the opposite pattern. Cell size would increase exponentially as a function of temperature. So we have some evidence for these assumptions, but they are not extremely robust. So this suggests that more investigations on how membrane constraints can affect cell size could provide us more information about this mechanism, about how robust this mechanism is. And finally, what I wanted to show is that even if we assume we make these two assumptions and cell size decreases as a function of temperature, when the cell is near death, is struggling a lot to survive, it gets bigger again. And why is that happening, right? Well, So I wanted to say our model predicts that the cells get bigger at very high temperatures. And that's something contradictory, but actually recent studies, this study by Elisa Shawn has evolved diatoms at different temperatures, and they found that after adaptation, the cells could actually get bigger in the higher temperature treatments. So this begs the question if the cells will find a way to actually circumvent this size constraint as temperatures increase and perhaps get bigger. So in our model, this is what explains these changes. So when we assume that transporters have lower temperature sensitivity and that cells can pack more lipids as a function of temperature, you have an increase on the investment of these two processes. And that's why we see a decrease in cell size. But as temperatures get very high, then the cell needs to divest all resources from transporters and lipids and invest everything that it can on energy acquisition. And that's why cell size gets bigger again. All right, maybe I'm running out of time. But I would like to also share with you some exciting results about the metabolic switch at high temperatures. So with this model, we could test the trade-off between glycolysis and lipid degradation. And this trade-off is basically included in the model because one pathway has a higher energy yield compared to the other, but the more energetic pathway has a higher proteomic cost. So we would like to see if we could predict this switch with this model. And what we see here, as expected by our hypothesis, is that at lower temperatures, the cell would invest in glycolysis. So there is low metabolic demand here, and the cell can go along with a pathway that has a lower proteomic cost, but also gives us a lower energy yield. However, as temperatures increase, the cell switches and obtain energy to pay off the dark respiration costs from lipid degradation. So what we are showing here is that even if this pathway has a higher proteomic cost, it can be advantageous at higher temperatures due to this high energetic yield. And also, space constraints can also play a role in this trade-off, since lipids can take up less space compared to carbohydrates. So this switch is something that has been more discussed in the literature recently, and especially in the biofuel industry, where they are more focused on producing a lot of lipids with these cells. So we are excited to see that lipid metabolism maybe play a more important role on phytoplankton responses to temperature than previously expected. So with that, I come to the conclusions of this work. I hope that I have convinced you that we could gain new mechanistic understanding by doing this exercise. And with this model, we found that the cells would invest on photosystems to meet Robisco demands at high temperature, and on repair proteins and respiration to meet the high metabolic costs of the cell, divesting resources from growth. And we also explored how membrane and proteomic constraints could explain changes in cell size due to warming, and that maybe cell size don't need to get always smaller. And a trade-off between proteomic cost and energy yield would favor lipid degradation over glycolysis at high temperatures. And with that, I would just leave this question here, which is something that we have been thinking about and working, is that if phototrophs could adapt to warming by overcoming any of these predicted constraints. And yes, thank you for listening. And now I will take any questions. Oh, I can't hear you, Steph. I don't know.
I think you guys are on mute. We can't hear you.
All right. How about now?
Yes.
Okay. Thank you very much, Suzana. That seems like an incredibly powerful framework, and I'm really excited to see more work come from it. I think David has a question.
Thank you, Heidi.
Yeah, I completely agree. That was absolutely beautiful presentation. The system is awesome. I think what I really like about it is that you're making predictions with the model that seem highly testable, which is one of the really nice ways that you can apply these kinds of models. I guess one kind of comment and question, having looked at some of the predictions of these kinds of models myself, one thing that I find often curious is that the predictions they make about reallocations in the proteome seem to over-predict what some of the proteomic data suggests. Mm. So they seem to predict quite large swings, like you were showing sort of like 50% of the proteome kind of reallocating, and it seems like that often the data is a bit more constitutive than that, and I don't really have a good handle on why that is. I'd be curious. Oh, well, sorry, I should clarify. I've seen that in the context of acclimation to nutrients and light, not to temperature. So I guess, sorry, this is a slightly meandering comment. I'd be curious to know if there is more flexibility in adaptation to temperature, and if you have any thoughts on that, and if you have any plans to try and sort of evaluate this in the context of some proteomic data, it'd be super cool. Thanks.
Well, David, you touched on a point that Naomi and I are super excited about, which is trying to collaborate with some group and get some proteomic data to actually check these predictions. That would be awesome. That's even proteomic data would be like ideal, right? But even looking at these transcriptomic studies, it was very exciting to see these patterns of up or down regulation of different pathways. So one answer. Like when we think about temperature changes, there are processes that happen very, very fast, right? Like the unfolding of proteins and the repair. This is much faster than other reactions. So this is just a guess because I never look at this kind of data, proteomic data. But my guess would be that you would see very quick changes on investment on these repair proteins and photosystems. Now you left me wondering if this could also be an issue of just because we have very simple model, right? We are just considering a few pathways but in reality there are many more. So I don't know if this could be like an artifact when you look at the real data for why you see smaller changes. But this is just a guess from someone that have never looked at this data. So sorry, I might not be the best to answer this question.
Maybe I have a related question maybe about whether, so a ton of temperature variation happens on the planet now, right? In temperate zones, the seasonality is huge. And can your framework, have you thought about whether it can reproduce observations under those temperature variations? Would that be helpful to evaluate the mechanisms?
I think it would be super helpful, definitely. This is something that I was also looking at the literature. There are some studies that compared warm and cold adapted species, including one of the transcriptomic studies that I cited here from the Liang et al. 2019. And I think this is super cool and something that I was interested on using this kind of data for this model is that the mechanisms that we are talking about here, they are both present for cold and warm adapted, but the magnitude of the proteome change, maybe of what David was telling about too, it could be different because cold adapted species, they will have one kind of strategy to deal with that very cold environment, right? So maybe they need to upregulate lipid synthesis and they don't need to care much about lipid degradation. So when they move to a warmer temperature, then they might need to downregulate this pathway of synthesis, but not really lipid degradation, while the opposite would be expected for warm species. So I guess you touched on an interesting point. I think this would be an interesting data set to test these model predictions.
Who wants to start the proteomics time series on the Northeast US Shelf LTR site, right? That would be so cool. Yeah.
Maybe David?
Hi, this is awesome work, and I'm going to have to stare at these slides for a few more hours to get a feel for what you're doing here, but it's really cool. My question is whether your model predicts changes in the abundance of carbon storage molecules across temperatures, so more or less lipids and or carbohydrates in colder versus warmer environments. And sorry if you've already covered this.
No, that's actually a super important point. So this model we are solving, it's not a dynamic model. It's that we are solving this equation that's steady state, and we do simulate like the internal pool of carbon as carbohydrates and the lipids in the membrane. But when it comes to storage, we need to be a bit careful here because we cannot simulate the storage when assuming that these cells are optimized, right? Like acclimated at a certain temperature. Because if you don't have like changes in the environment, you wouldn't have a reason to store. So something that would be exciting is actually to put this in the context of a dynamic system like the like DL cycles to see these changes in storage as a function of the environment, and then you could simulate that. But here we don't explicitly simulate storage pools.
Cool. Thank you. I agree. That would be cool to do.
Mm-hmm. It's more like what we are doing.
Yeah, hopefully you can see and hear me, Ariana. Amazing work. I just was wondering about when you said that at the highest temperatures, you would see increases in cell size again. Whether or not when you see that empirically, you mentioned that it was near death in some cases, whether you're still seeing higher growth rates at that point when the cell sizes increase back, or whether that's the growth rate trend has also shifted at that point.
That's a very great question. So the study that I showed was from Elisa Sean work. They adapted diatoms at different temperatures, and after adaptation, they could become bigger, and they were actually growing well. If I recall correctly, they were not near death on that study because they were adapted to that temperature. In our model, yes, it's very close to cell death, and their growth rates are getting lower and lower. So something that is interesting to think about here, like Elisa did an evolution experimental study, right? But when you try to check the acclimated response in short-term experiments, these might be very hard, right? Because if you push your cell at very high temperatures, maybe they would just die out, and it would be difficult to make any observations. So that's something that we were wondering when we saw these modeling results. Like, is this something that would be possible to see empirically? Or maybe we are just missing it because it's just too hard to do in the lab. I hope this answers your question, Ariana.
Hi, Suzana. This is Jessica. This might be a bit of a naive question, but from other optimization models, such as maybe David Tommy's from 2013, I kind of got the impression that the investment towards repair was relatively a small investment. And I was wondering from your results here, what the difference would be in growth rates or respiration rates if you didn't include repair.
Okay, so I have done different model runs, and when I started running this model, I was just including like the damage without repair. I think that's what you were saying, and then it's just an extra cost. The cell doesn't need to invest in repair, but at the same point, you have these damaged photosystems just accumulating in the cell, and they take up space. So actually, if you don't have repair, you would have in our model lower growth. But if you see it doesn't need to have a super high investment on repairs. I can go back there. Sorry, so many slides. But yeah, it would be interesting to chat more with David after. I would like to do that. But yeah, we tried, and then the difference would be lower growth rate and this space that is just accumulated. And we don't account for it in the model, but you can also think that damaged proteins, they also add up ROS, reactive oxygen species, and could add to stress.
Thanks.
Ken, maybe?
Ken, go ahead.
Yeah, okay, thanks. Yeah, it is very impressive, and I think you have opened the box for a lot of different explorations one can do with these models. So Camila Salar Pompey did a very similar study, but in a much less complicated model than this here, because she wanted to do it dynamically and not in steady state. But what she saw was that the result depended a lot on the conditions of resources, I mean, nutrients and lights. And I wonder whether you have looked at how that changes the outcome of the model here.
Yeah, we can definitely do that. And there is something that we are working on, especially changing nutrient concentrations. So this is something that we also wanted to explore with this model, because when people observe like cell size decreases in the ocean, right? And then usually they evoke also mechanisms of nutrient limitation to explain these patterns. So we were interested particularly here on trying to remove this effect to see what would happen. But I did run simulations in which, let's say, temperatures are decreasing and nutrients are decreasing as well. And then what you would see is a much harder growth environment. So the growth rate would decrease drastically. And yes, the cell needs to invest more and more on transporters because now nutrients are depleted in the system. But it's just, depending on how much hard you push, they just die out in this model. It's hard.
Yeah, of course, I was more thinking about how the temperature response was depending on, say, nutrient and light also. But yeah.
Well, we're just feeding into the discussion. You want one more question?
That was a fantastic model. I very much enjoyed the presentation. There's a lot more than I can also gather just from the short talk. But I was curious on the your Arrhenius response, which I'm guessing is affecting the reactions or the pathways in your model. Was it similar for all the pathways, or were you able to get data to try to change how the Arrhenius response for different pathways might happen?
Yeah, this is something that we have done a lot of sensitivity analysis. So I started considering the same Arrhenius function for all metabolic processes, right? And then if you assume that everything is equal and you don't assume damage, then everything will be constant, right? You see just exponential growth and the investment will not change. But then if you assume all the rest is equal as well in the model. So this was kind of where I started with this with playing with the sensitivity analysis. But then as you change the sensitivity, the temperature sensitivity of a given process, this will drive the relative investment. So for example, here we have strong evidence that photochemical reactions are temperature independent. So this is something that is very robust and we can do. So what we started thinking about nutrient transporters and if these would be less sensitive to temperature than other processes because of the changes in cell size. There is some evidence for that. And on this seminal work by John Raven and Richard Geider, maybe on the 80s, and they give a list of like the slope of these curves for different processes. And that's a good start point for us to try to play with this sensitivity. But you are right, there is large uncertainties, and it's something very important to understand the dynamics of the model.
Okay, let's thank Suzana again. That was great discussion.
Thank you.